I want you to imagine trying to draw a flawlessly accurate map of a bustling, peaceful city.
Okay, I'm picturing it.
Right. You want to understand its normal, everyday traffic patterns, where people work, where they buy their groceries, how the neighborhoods interact.
Sure, the day-to-day stuff.
Exactly. But there's this massive catch. The only time you're actually allowed to observe this city is right after a catastrophic earthquake.
Oh, wow.
Or, alternatively, you can only look at a neighborhood that's sitting right next door to a raging, uncontrollable fire.
That sounds less than ideal.
Yeah, right. So how accurate do you honestly think your map of normal would be under those conditions?
I mean, it would be completely distorted. You wouldn't be mapping daily life at all. You'd just be mapping panic, crisis management, and emergency response.
Exactly.
And what's fascinating here is that historically, in the world of biology, this is exactly how we have been mapping the human liver.
Which is wild to think about.
It really is. But today, we are diving into a groundbreaking 2026 Nature paper that just fundamentally changes that entire paradigm.
Yes, it does.
Because this research created the first high-resolution spatial atlas of a truly, definitively healthy human liver.
And that is the mission of this deep dive. We are going to explore the liver cell by cell, lobule by lobule.
Literally mapping the entire city.
Exactly. We're going to unpack how a healthy liver actually functions and discover why human biology is radically different from the lab mice we've relied on for decades.
Right.
And we're going to look at how individual cells communicate purely based on their microscopic geography.
So to truly appreciate the scale of this breakthrough, we first have to understand the historical hurdle, what we can call the tissue source conundrum.
The earthquake and the fire.
Exactly. For decades, human liver atlases relied heavily on two deeply flawed sources. The first is tissue from NDDs, or neurologically deceased donors.
Okay, let's unpack this. A neurologically deceased donor is someone whose organs are kept viable on life support prior to donation. But viable doesn't necessarily mean biologically neutral, does it?
Not at all. No. Livers from NDDs undergo severe metabolic distress before they are ever studied.
Because of the medical interventions.
Yeah, exactly. They experience prolonged mechanical ventilation, artificial non-enteral nutrition, and, most critically, ischemia.
Which is a lack of blood flow, right?
Yes, a severe lack of blood flow before the tissue is actually collected. And at a molecular level, this ischemia causes transcript-dependent RNA degradation.
Wait, let me make sure I'm getting the mechanism right here. RNA is basically the temporary blueprint a cell is actively reading to build proteins, right?
Precisely.
And because it's just temporary, it's highly unstable.
Yeah, exactly. Unlike DNA, which is locked away safely, RNA is inherently designed to be degraded quickly.
Oh, I see.
So when blood flow stops during ischemia, cellular enzymes called RNases start aggressively chewing up these RNA messages. The blueprints literally start falling apart.
Wow.
And studies show this ischemic degradation alone can explain up to 40% of the variability we see in RNA sequencing.
That is a massive margin of error.
It's huge.
So that's the earthquake scenario. But what about the other historical source? You mentioned mapping a neighborhood near a raging fire. That's the adjacent normal tissue, right?
Yes, adjacent normal.
Right.
So often when a patient undergoes a liver resection to remove a tumor, surgeons will provide a piece of tissue taken about 5 centimeters away from the cancer.
Okay.
A pathologist looks at it under a microscope, sees no visible cancer cells, and declares it histologically normal.
So it looks fine.
It looks totally fine. But the transcriptomics, the actual gene expression, they tell a completely different story.
It's exactly like that neighborhood near a crime scene. I mean, the houses, the physical structure of the cells, they look completely fine from the outside.
Right.
But if you listen to the radio traffic, the mRNA, everyone inside is nervously whispering.
That is a perfect analogy. The researchers proved this by using an entirely new, incredibly rigorous cohort, 8 LHDs, or live human donors.
Oh, wow.
Yeah. These are intensely screened, perfectly healthy people donating a lobe of their liver.
Because the human liver regenerates, so this is actually possible.
Exactly. And when they compared these healthy LHDs to the adjacent normal tissue we used to rely on, the differences were just staggering.
Like what?
Well, the adjacent tissue had drastically lowered canonical liver functions. For example, ASS1, which is arginine synthase 1, was significantly downregulated.
Wait, what does ASS1 do normally?
It's a critical enzyme in the urea cycle that helps clear toxic ammonia from the body.
Oh, okay, so that was shutting down.
Yeah. And so was PCK2, which is a crucial gene for making glucose.
So the normal factory functions were literally shutting down. What was turning on instead?
Immune and stress responses. Specifically, calprotectin genes like S1000A8 and S1000A9 were highly elevated.
Okay, so what are those?
These are basically alarm bell proteins. The tissue was bracing for impact, fundamentally altered by the systemic stress of the nearby tumor.
This completely resets the foundation of what we thought we knew.
It really does.
So if I'm synthesizing this correctly to make sure I'm tracking, let's do a quick recap.
Yep.
Number 1, dead or diseased livers massively alter their RNA due to lack of blood flow and medical interventions.
Yep.
Number 2, tissue located next to tumors is functionally stressed and panicking, even if it physically looks perfectly healthy under a microscope.
Exactly.
And number 3, these live human donors provide the very first biologically true baseline for mapping human tissue.
That's an excellent summary. And it leads to a question for you, the listener. Think about your own field of work, your research, or even your daily routines. Are the baselines you rely on actually as neutral and healthy as you think they are? Or are you, too, unknowingly mapping the aftermath of an earthquake?
That is a phenomenal question to hold on to. Now, if we actually have a truly healthy map, we need to talk about the geography of the liver itself. We need to decode the architecture to understand how it functions.
Right, because the liver is highly rigorously organized. The main parenchymal cells, the heavy lifters, are called hepatocytes.
Okay, hepatocytes.
And they are arranged into these microscopic hexagonal structures called lobules, each about 0.5 millimeter to 1 millimeter wide.
So tiny little hexagons.
Yeah. Now picture this hexagon. Blood flows into the lobule from the outer corners, which are called the portal veins.
Got it.
This blood is extremely rich with oxygen and fresh nutrients straight from the gut. And it flows inward toward the very center of the hexagon, draining into the central vein.
This creates a really fascinating dynamic. It's like a river flowing through a manufacturing town, right?
Exactly like that.
The factories located upstream, in that portal zone at the outer edge, they get the absolute freshest materials and the most oxygen.
Right.
And then the factories located downstream, in the central zone, they have to adapt to use whatever depleted resources are left over.
Yeah, they get the leftovers.
This spatial segregation of duties, based entirely on blood flow, is called liver zonation.
Exactly. And the technology stack the researchers deployed to map this zonation is just staggering.
I bet. What did they use?
Well, first they use Visium and Visium HD. This is spatial transcriptomics.
Okay, how does that work?
Imagine taking a micro-thin slice of liver tissue and placing it on a specialized glass slide covered in a microscopic grid of millions of unique DNA barcodes.
Oh, wow. So when you dissolve the tissue on the slide, the RNA from the cells drops straight down and sticks to the specific barcode right underneath it.
Yes, exactly. You sequence the RNA, read the attached barcode, and suddenly you know exactly where that specific gene was active in the geographic landscape of the lobule.
That is incredibly precise.
It is. But they didn't stop there. They coupled this with MERFISH.
Multiplexed error-robust fluorescence in situ hybridization. That is a massive mouthful.
It is a mouthful, yeah. But the mechanism is beautiful. They assign unique fluorescent color codes to hundreds of specific RNA molecules.
Okay, color coding the RNA.
Right. So under a microscope, they could literally watch like 500 different genes light up like a starry night sky inside intact individual cells.
That sounds...
And the error-robust part is key. Biological tissue is messy. Things glow randomly. So MERFISH uses complex error-checking barcode sequences, so the algorithm doesn't mistake a random microscopic spark for a true genetic signal.
That's super smart. And to top it off, they use the PhenoCycler, right?
Yeah.
Which is this advanced imaging system that looks at 48 different protein markers simultaneously on the exact same slice of tissue.
Exactly. By layering the RNA data, the spatial barcodes, and the protein imaging, they mapped exactly which factories were active upstream versus downstream.
Let me lock this in for us with a quick recap. Number 1, liver cells live in tiny hexagonal neighborhoods called lobules.
Right.
Number 2, the inward flow of blood creates a massive gradient of oxygen and nutrients. Number 3, because of this gradient, cells completely change their function based on their geographic location, and that's zonation.
Yes.
And number 4, cutting-edge spatial technologies like Visium HD and MERFISH allow us to map these microscopic gradients with pinpoint accuracy.
Which brings up another question for you. If you were a cell in this highly organized city, would you want to live in the highly resourced, oxygen-rich portal zone? Or would you prefer the highly specialized, highly adaptive environment of the central zone downstream?
Honestly, I think it'd be an upstream portal zone cell. I really like having first dibs on the resources.
Fair enough.
But here's where it gets really interesting. Because if you asked a mouse that same question, you'd get a totally different biological answer than if you asked a human.
Oh, absolutely.
Because the researchers didn't just map humans, right? They created spatial atlases for mice, domestic pigs, wild boars, and cows to compare.
Yes. And this interspecies comparison yielded the most profound reveal of the entire paper. Human livers feature a massive fundamental pericentral shift.
A pericentral shift? What does that mean?
Well, in mice and in those larger mammals, the most crucial metabolic liver functions happen in the periportal zone, the upstream factories.
Right, where the fresh blood comes in.
Exactly. We're talking about the urea cycle enzymes that clear ammonia, like ASL, AAG, and OTC.
Okay.
We're also talking about the critical glucose transporter, GLUT2, which is encoded by the SLC2A2 gene. In lab mice, all of these are stationed right where the fresh blood enters.
But in humans, we completely flipped the script.
We really did.
All of those heavy-lifting metabolic genes are aggressively shifted downstream to the pericentral zone.
Yeah.
So if we use our town analogy, imagine human central factories aggressively hoarding all the glucose transporters and urea cycle tools, taking the standard mammalian blueprint and turning it completely inside out.
That's a great way to picture it. And this shift helps explain a long-standing mystery called the glucose paradox.
Oh, I've heard of this.
Yeah. So in the human liver, glycogen, which is your stored sugar, is replenished directly from glucose in that pericentral zone. But in the upstream periportal zone, it's actually replenished from lactate.
Let me pause you there. Lactate, like the byproduct of muscle fatigue.
Yes, exactly that lactate. And we can see this geographically because of the spatial expression of two specific genes, LDHB and LDHA.
Okay, what do they do?
LDHB, which converts lactate into pyruvate to be used for energy, is heavily expressed upstream.
Yeah.
Downstream, the cells are just using pure glucose.
But why the massive evolutionary shift? I mean, is human biology just built with a totally different architectural blueprint than the rest of the animal kingdom? Did we evolve a completely new signaling system?
That's the logical assumption, right? But this raises an important question, and the answer is surprisingly no.
Oh, really?
Yeah. The fundamental architect, the master regulator of liver zonation, is a highly conserved cellular communication network called the WNT signaling pathway.
The WNT pathway.
Right. This involves morphogen signals like WNT2 and RSPO3, binding to a receptor on the cells called LGR5.
Wait, a morphogen signal, meaning it's a chemical that creates a physical gradient.
Exactly. The WNT signal is highly concentrated at the central vein and diffuses outward, creating a literal chemical slope that tells the cells where they are geographically.
Oh, I see.
And what's fascinating here is that the researchers found this WNT blueprint is perfectly conserved pericentrally across humans, mice, pigs, and cows. The chemical slope is identical.
So the foreman handing out the zoning permits is standing in the exact same spot across all species. But the human cells are looking at the permit and choosing to build entirely different factories.
You nailed it. The WNT signal tells them where they are, but not what to do.
Wow.
The researchers hypothesize this shift is likely driven by our unique human metabolic rates, our distinctly different diets, and uniquely shaped nutrient gradients that just force our cells to adapt differently than a cow or a lab mouse over evolutionary time.
Let's do a quick synthesis of this evolutionary twist. Number one, human liver zonation is radically different from mice and other large mammals.
Right.
Number two, key metabolic functions like glucose transport and the urea cycle shift heavily to the downstream center in humans. And number three, the WNT signaling pathway remains the universal architectural blueprint across species, meaning environmental and metabolic pressures drove the human shift, not a change in the core biological wiring.
And here's a critical question for you. Given this massive geographic difference in how our human cells function, how much should we continue to rely on lab mice to test human liver metabolism and drug toxicity?
That is a very good question. It certainly makes you view pharmaceutical clinical trials in a whole new light.
Absolutely.
Now, if the main factory workers, the hepatocytes, are moving all this infrastructure around, who is building the roads? I mean, who is facilitating all of this? We need to talk about the supporting cast, the non-parenchymal cells, or NPCs.
Right, because the liver isn't just hepatocytes. You have LSCCs, liver sinusoidals, and endothelial cells. Think of these as the structural pipes lining the blood vessels.
Okay, LSCCs are the pipes.
Then you have fibroblasts, which are the structural cells that build the collagen scaffolding of the organ.
Got it.
And crucially, you have Kupffer cells, which are the resident macrophages. essentially the liver's immune system janitors.
So to study these supporting cells, the researchers used a technique called snRNA-seq, or single-nucleus RNA sequencing.
Yes, they did.
Now let me challenge you on this methodology because it sounds crazy. To get single nuclei, they had to totally dissolve the human tissue to isolate 67,000 individual nuclei, right?
Yeah, that's the process.
But wait, if you completely dissolve the tissue, you destroy the map. You lose all the geography. That sounds like unmixing a smoothie. How could they possibly know where these floating cells originally lived with 2-micrometer accuracy?
I mean, it does sound exactly like unmixing a smoothie. And this is where their computational brilliance comes in. They used advanced machine learning.
Of course, machine learning.
Specifically, an algorithm called Stardust. Think of Stardust as a hyper-intelligent digital cookie cutter. They trained it to look at standard H&E stained pathology images and visually identify the exact borders of every single cell.
Okay, so we have a visual map of the borders.
Right. Then they combine that visual map with their Vision HD spatial transcriptomic data.
Right.
Remember that grid of DNA barcodes we talked about earlier?
Right.
By training a random forest classifier algorithm on both datasets, the computer learned the unique genetic accent of each geographic zone.
Oh, wow.
Yeah, so it was then able to digitally take those 67,000 floating dissolved nuclei, listen to their genetic accent, and assign them back to specific 2x2 micrometer coordinates on the physical map.
They literally digitally rebuilt the dissolved city.
Exactly.
And by doing that, they could listen in on the cellular chatter. They mapped exactly who was talking to who across the structural void.
And they found incredible, highly specific crosstalk. For example, in that downstream pericentral zone, the LSECs, the blood vessel pipes, are the ones actively producing those WNT2 signals, sending them directly to specific receptors called FZD5 on the hepatocytes.
Okay.
Meanwhile, the structural fibroblasts are broadcasting signals like BMP9, encoded by the GDF2 gene, and BMP10 to physically guide the behavior of both the hepatocytes and the blood vessels.
It's a beautifully choreographed dance, but my absolute favorite discovery here is the Kupffer mystery.
Oh, yes.
Remember, these are the immune janitors. In mice, these janitors mostly hang out in the periportal zone, right? Upstream, right where the fresh blood and potential gut pathogens first enter the liver.
Right, standing guard at the gate.
But in humans, they are shifted downstream to the pericentral zone.
Which makes perfect physiological sense when you remember the pericentral shift we just talked about.
Oh, right.
Human downstream cells have taken on the massive, heavy metabolic lifting. And that heavy lifting, clearing ammonia, processing glucose, it generates significantly more cellular debris and requires much higher cell turnover.
I see.
So the immune janitors simply packed up and moved their break room to the zone where the most microscopic trash is being generated.
Form follows function perfectly. Here is your quick recap for the supporting cast. Number 1, the liver relies on a highly complex network of supporting cells like LSECs, fibroblasts, and Kupffer cells to function. Number 2, researchers successfully used machine learning algorithms to act as a digital cookie cutter, fusing floating single-cell data back onto high-resolution spatial maps.
Yes.
Number 3, cells communicate via highly specific geographic chemical signals depending on their zone. And number 4, human immune janitors migrate to the center of a lobule specifically to handle our uniquely human high metabolic waste output.
And my question for you is this. Think of the communication networks in your own life, your team, or your organization. Do you have specific zones where information flows better than others? And more importantly, are your problem solvers located where the actual heavy lifting is happening?
A fantastic organizational lesson derived straight from our biology.
Truly.
Finally, we need to bridge this healthy baseline into the clinical reality. What happens when things start to go wrong? The researchers didn't just look at perfectly pristine tissue, they also mapped early steatosis.
Right, the microscopic stepping stones of fatty liver disease.
Oh.
And this is a vital inclusion. When a live donor gives a liver graft, transplant surgeons consider the tissue perfectly usable and clinically healthy, even if it contains up to 20% steatosis, or fat accumulation.
Wait, really? 20% fat is still considered healthy?
Yeah, it's a very common baseline in modern humans.
Interesting. So to study this incredibly early stage of fat accumulation, the researchers once again unleashed machine learning. They trained an algorithm to look at the microscopic pixels on the images and actually count, measure, and precisely locate the lipid droplets, the little bubbles of fat, forming inside the lobules.
Exactly. And by precisely tracking the physical volume of fat in each specific 2 by 2 micrometer square, they could overlay their transcriptomic data.
Oh, that's brilliant.
They didn't just see the fat, they could see the exact dynamic gene expression changes occurring at the exact moment the lipid content began to increase.
They caught the cells in the act of desperately compensating.
Yes, they did.
As those lipid droplets physically grew and began to crowd the inside of the cell, the researchers watched a massive genetic divergence. The nucleus of the cell started downregulating nuclear-encoded mitochondrial proteins.
Right, so if we connect this to the bigger picture, the fat is literally suffocating the cellular engine. The main headquarters, the nucleus, sees the energy crisis and stops sending blueprints to build new mitochondrial engines, basically trying to slow metabolism down.
But simultaneously, the researchers saw a massive compensatory rise in mitochondria-encoded transcript.
Exactly. Because mitochondria possess their own independent DNA, they don't have to wait for the nucleus.
Oh, right.
The local mitochondria sense the physical crowding from the fat, they panic, and they start cranking up their own internal transcription, desperately trying to keep the engines running despite the toxic environment.
That is incredible.
It gives us a flawless molecular timeline of disease progression before the disease is even clinically relevant.
Let's do our final synthesis for this clinical application. Number 1, clinically healthy transplant livers can safely contain early microscopic signs of fat.
Right.
Number 2, machine learning successfully tracked and quantified these fat droplets across the geographic landscape. And number 3, we can now witness the exact genetic shifts, the panic between the nucleus and the mitochondria that happen the exact moment a liver cell begins to become fatty.
Which leaves us with this final question for this segment. If we can map the earliest genetic whispers of disease at a microscopic subcellular level, what does this mean for the future of preventative medicine? Could we intervene pharmaceutically before the physical damage is even visible?
So what does this all mean? When we zoom out and look at this entire deep dive, this paper isn't just a highly detailed map with pretty fluorescent colors.
No, not at all.
It is a completely new foundation for human biology. By proving that our old, adjacent, normal, and brain-dead donor samples were inherently flawed, and by definitively proving that human livers operate fundamentally differently from the lab animals we've relied on for decades, this paper effectively resets the clock on liver research.
I couldn't agree more. The ultimate goal here is precision medicine, and precision medicine fundamentally requires a precise baseline.
Exactly.
Without understanding the healthy, highly organized spatial architecture of the human liver, treating complex, spatially driven diseases like autoimmune hepatitis, cholangitis, or fibrosis is like trying to fix a highly complex engine without a schematic.
Right.
Now, thanks to spatial transcriptomics and machine learning, we finally have the schematic.
It's truly incredible, and it brings us right back to our very first analogy. For decades, we were trying to map the city after the earthquake, or while looking at the mouse-sized model town down the road. Now, we can see the human traffic flowing, the downstream factories humming, and the immune janitors cleaning up in real time, exactly as nature intended. Which leaves me with one final thought for you to ponder.
Let's hear it.
If our liver, this massive, essential, multitasking organ, operates on such deeply specific, microscopic geographic gradients, what other organs in the human body are hiding massive functional secrets based purely on their microscopic geography? I invite you to apply this concept of zonation to the natural or artificial systems around you. Sometimes where something happens is just as important as what is happening.